Ladies and gentlemen, I'm delighted to welcome you all to the Swedish residence for today's discussion on protein science. My name is Andreas from Beckerath. I'm Deputy Head of the Swedish Embassy here in London. Before I hand over the word to Daniel Mahony, our chair for today's interesting discussion, let me take this opportunity to say a few brief words on Sweden as a country of research and development. We Swedes are very proud of our heritage of innovations and inventions. Most Swedish school children learn about Swedes that developed and invented dynamite, the zipper, the safety matches, and the adjustable wrench. But throughout the years, one sector in more modern times that has been developing extremely strong is the sector of life science, which has contributed to innovations such as the cardiac pacemaker and the artificial kidney. And I think there are many different reasons why we have been so successful in terms of research and development in Sweden. One important aspect is, of course, that we invest 4% of our GDP in research and development, which is one of the highest rate in the world. Personally, a contributing factor, I believe, is that Swedes tend to be very open-minded when it comes to trying new ideas and new technology. It's enough to stroll the streets in Stockholm in the spring now, and you will see all the young people very eager to try out the latest fashion and to buy the latest technology products. And the third reason I believe that has been very important is the important collaboration between the academic sector and the corporate sector, and also the ability to work across sections of science. And one region that has been particularly successful when it comes to bringing together successful businesses and corporate organizations with academic environment has been Stockholm-Uppsala region, and particularly in the life science sector. They have been able to bring together big companies such as GE Healthcare and AstraZeneca, together with world-class institutions such as Karolinska Institutet and Uppsala University. They host more than 25,000 specialists, boast more than 500 companies and 300 larger research projects. And last but not least, it's the home of the Nobel Prize. But one person who knows a great deal more about life science and also, I think, about the Stockholm-Uppsala region is our host today, Daniel Mahony. Daniel has more than 12 years experience of investments in the healthcare sector. He's presently with Polar Capital and has been before with Morgan Stanley, and has also worked 7 years as a scientific researcher in California, has a PhD from Cambridge. Daniel, the floor is yours.
When I finished my PhD, I went to California, which is a beautiful, hot place. And while everyone around me was busily beginning to sequence the genome, I spent 4 years in a cold room trying to become a biochemist, packing my own chromatography columns. It certainly wasn't the age of protein science then. And I was so successful at that I moved to Wall Street in 1998 and gave up science completely. I have a scientific background, I understand a fair amount about biochemistry, but I also understand this session particularly is about hype and reality. And one of the difficulties when you're an investor, and also I suppose in your job as journalists, is what's real and what is just being made up and being exaggerated. And I think that when we think about reality, what does that mean? Well, for me as an investor is, when is this particular technology actually going to start making money? I think the other sense of reality is, when are these technologies actually going to have an impact on society? And I think those are two sort of maybe related questions, but they're separate questions. And hopefully those are the sorts of things we can talk about today. And I think when you think about protein sciences, it's sort of, and that impact on society, I suppose when you think about the value chain, you can think about what's its impact in research? How can we use that to discover better drugs more quickly, etc.? We can think about that in diagnosing disease, which I think is a very important thing. And then ultimately, it ends up in the area of therapeutics, what drugs, protein drugs, that can actually treat people. And so I think the question we'd like to talk about today is it hyperreality? Are we there yet? Are we getting there? And I think what we'd like to create is quite an interactive session. Speak up, ask questions. I think we're very lucky to have 4 experts here in different areas of that value chain who are quite happy to answer anything, apparently. So why don't we kick off, and I think everyone's going to just give a quick 1-minute introduction, and then we can start a discussion. Do you want to go first, Lotta?
Yes, so I'm Lotta Ljungqvist. I'm heading Research and Development for GE Healthcare Life Sciences. We cover a very broad range of products, developing technologies and hardware and software and chemistry-based products for the biopharma industry and also for academic research and discovery units. The main facility is located in Uppsala, where we have about 1,200 employees, and the majority of the R&D is located in Uppsala. Very strong in purification of protein-based drugs. We have, our products are used in roughly 90–95% of all the protein-based drugs that are manufactured. Use at least one of our products, so we have a very strong recognition of our technology in the biopharma industry. So I guess that's the brief version of who I am.
My name is Christian Tryggvason. I'm the CEO of Biolamina. Biolamina has invented or found recombinant proteins that you can use to culture stem cells and other differentiated cells on, which has not been, or it has been possible before, but we have made the process much easier, much better. I also worked previously for a company called Atlas Antibodies that is famous for basically commercializing all the proteins that come from the human proteome project in the Stockholm-Uppsala region. Yeah.
Yep, I'm Gunnar von Heine. I come from Stockholm University, and I represent basic science here. I think you can tell that we've been very carefully choosing our dress code to make it easy for you to identify who is who. So I'm a protein chemist or biochemist. I work on membrane proteins. I'll come back to that. I've started, I've been in this field for about 35 years, I think, since 1975 roughly. So I've seen a lot of developments. During this time, I've started a couple of centers. I started Stockholm Bioinformatics Center about 10–12 years ago to bring the bioinformatics expertise into life science in the Stockholm-Uppsala region. More recently, I've started a center for biomembrane research, which is what I'm directing right now. I'm also vice director of something called Science for Life Laboratories, which is a new initiative in the Stockholm-Uppsala region, where we're putting all the high throughput technologies in life science into a common environment. And the two of us are actually collaborating closely in this project. So that's actually a very big undertaking and funded by the Swedish government. And finally, just to get it off the table, I used to be, up until a year and a half ago, I was chairman of the Nobel Committee for Chemistry. So I know a little bit about the Nobel Prize as well.
Okay, well, I'm not sure about my dress code, but I'm maybe somewhat intermediary. I'm an MD Ph.D. and professor of molecular medicine. I have a medical interest, but I view myself more as an engineer, perhaps. So what you'll hear later is our work on trying to develop better ways of analyzing the proteome. I have a history with more genomics-oriented technology development, and we've been spinning out a number of companies around our technologies, also licensing technologies to the major biotech companies, Life Technology and Affymetrix, and once upon a time GE also, or the predecessor of GE. So we are working on what I consider more basic research than basic research, for the tools that you need for basic research, but we also are commercializing them. I am, like you, a co-director of the Science for Life laboratory. I don't know how much you'll hear about that, but that's a very big effort in the Stockholm-Uppsala region and Sweden generally.
So perhaps I'll kick off. So when I think about proteins and sort of impact on treatment, etc., I think of therapeutic antibodies. So when I turn to you, Lotta, I mean, particularly when we think about how antibodies are purified, obviously GE Healthcare has been helping the industry for many years. And if I look at therapeutic antibodies, probably I think by 2015, once we get through the current round of patent expirations, therapeutic antibodies are going to be the best-selling drugs. You know, I think 4 or 5 of the top 10 selling drugs will all be therapeutic antibodies. So perhaps put a bit of a historical context around that. What has Sweden done to help facilitate that? What are you going to do going forward? Because that seems to me that's a reality that is today. What can you do to help that part of the industry?
It's definitely a reality today, I think, because what we see is we provide technology that is used to purify these monoclonal antibodies that are then used as a therapeutic. And you see hundreds of them in clinical development. You see a couple of them are also already commercialized drugs. And we see with the technology we provide, we have helped to facilitate, to streamline the process to manufacture, to reduce development times, and to reduce the manufacturing costs. And I think that's a way to sort of make these products accessible for a broader population. It's protein-based drugs in the maybe 10, 20 years ago used to be the odd ones, the tricky ones. It's now become an everyday technique. It's easy to use, and it also facilitates this new technology to translate to population health. So I definitely think it's real. And we see that with all the products we provide, the sales of those products and the growth of that also translates into sort of growth in all regions. So it's moving over to also like India and China. They're now using and adopting this protein science. So it's a very well recognized technology, and it's definitely here. I couldn't say no to the question if it's if it's real. It's definitely real for us.